Swayam Prabha. Digital India. Educated India.
In this lecture, we are going to discuss in detail the optics of a compound microscope. Now, a compound microscope, unlike a simple microscope, has two lens systems. Now, if you compare a simple lens, it is made of a single lens which can be hold or which can be an object holder and could be mounted on a support. For example, if I show you in this on this screen, then you can have a simple holder which can be holding this lens, and this lens could be moved up and down here to which can be moved up and down to adjust the focus, and magnification can be done on this lens. Now, as we see that compound microscope is little different from here, as I will show you here. Now, in compound microscope, rather than one, there are usually two magnifying systems, one in tandem, one defined by objective and other defined by the eyepiece. Now, this avoids practical problem of making lens of a very short focal length. If you see in this figure, the object is placed just after the focal length or more than the one focal length of the lens. The objective forms the image, which is real inverted image. If you could recall, we have discussed this in the last lecture. Now, this real inverted image will act as an object for the eyepiece, and the eyepiece will form another image which will act as an object for the eyes, and finally, the image enlarged image will be formed on the retina of the eyes, which will be seen as a virtual image, or which will be seen as inverted image, as we will discuss it in later. Now, a compound microscope, if you see, this is a very simple diagram of a compound microscope, and we will discuss all different parts of compound microscope here. The compound microscopes can be of different kinds. They could be simple with different optics like fluorescence optics, it could be a phase contrast microscope, it could be a stereo microscope, or it could even be an inverted microscope, and like so on. Now, here if you see as we go along with different components of the microscope, first thing is the light source, which is uniform bright light source is placed either externally like shown here, or it could be mounted in the base of the microscope right below here. Now, there is another lens system, condenser lens system, which collects the light from the light source, and this is the condenser light passing from the condenser will be illuminating the object in a very small cone of light, which is focused on a very small area, and all stray light or extra light is being avoided by the apertures and iris, which are placed at different places. The object which is illuminated here, which is at the specimen stage, will the light or the light from condenser will be illuminating the object, and the light from object or specimen will pass on to the objective lens. Now, there will be two kinds of lights here, one which is not altered by the specimen, and another will be altered by the specimen. Now, one which is not altered, that is not diffracted, will be forming the which is not altered will be forming the background, and one which is diffracted will be focused by the objective to form the final image at the intermediate image plane. Now, after objective, there is a tube here which connects the objective to the eyepiece. The objective forms an image which will be real and inverted image, because the specimen is placed just one focal length away, little farther from one focal length from the specimen. So, a real inverted image is formed in here. The inverted image formed acts as an object, and the eyepiece which is connected here at the other end of the mechanical tube will be forming will be making image at virtual image, which is like here. If you have recalled that when you put your object below one focal length, then a virtual image is formed, which is formed at the base of the microscope and or in the mechanical tube, and this will be acting as an object for the eyes, and eyes will be the eye lens will form the final image on the retina, and which will be viewed on the retina as an enlarged or magnified image, because it occupies larger area on the eyes. Now, let us discuss some of the parts here to understand the microscope. Now, here condenser lens, which is we will be discussing it in detail also. Condenser lenses forms a very fine focused light onto the specimen, which is to avoid any stray or light which could form a diffused image. Now, always there is a diffusing screen put before the light source, after the light source and before the condenser, so that the image of light source is not formed here. If you see here objectives here, two objectives, there are these objective lenses which are all corrected. I could if you can recall, all the lenses including condenser, objective and eyepiece are corrected for various aberrations. Now, here if you see these two lenses which are objective lenses, here more than two lenses could be put in, which could go up to 5. Now, these objective lenses are attached to a rotating nose piece which is threaded in here, and this is connected to a tube length which is finite tube length, and on the other end of the tube length is the eyepiece. Now, here these objectives could be, this nose piece could be rotated, and the objective of different magnification, it could be 10x, it could be 20x, it could be 40x or 60x magnification objectives could be used for magnification as per the application. And most of the time, most of the advanced microscopes has will be parfocal, or in other words, the specimen will not lose focus when you are changing the objective by rotating the nose piece. So, that is a very important thing in here. Now, mechanical tube length is a fixed tube length, and the problem, there are two kinds of optical systems here. One is finite optical system, as I will show you in the next slide. Another is infinity corrected optic systems. It is very difficult to put in auxiliary components in finite optical system or fixed tube length, because of problems of correct image formation or sharp image formation due to aberrations and other problems. But infinity corrected optics, there will be option to put in the auxiliary components like prism or polarizer or other things, so that you can be and without any problems in the image here. Now, here if you see the objective, the mechanical tube length will be defined from the nose piece where it is threaded till where eyepiece is attached. Now, here if you see, if you consider eyepieces, there could be different arrangements here. It could be monocular, it could be binocular, or it could be trinocular port. Monocular means a single pair of eyepieces attached in here. Binocular means two eyepieces are attached, and trinocular means the three eyepieces are attached in here. Now, eyepieces could be arranged in different fashion. Now, here it could be monocular or binocular eyepiece or a trinocular eyepiece, or we also call it as a trinocular port. Now, most of the microscopes will have a inclined viewing for easy viewing, direct viewing here through eyes. If you have upright eyepieces, then it is very hard to view, like you have to stand and you cannot be easily viewing that. So, for inclined viewing, like I will show you on this screen, the eyepieces or the mechanical tube lengths are inclined at an angle, so that you can easily see the you can easily observe the image in the compound microscope. So, if I can say, if it is a binocular or a trinocular port, then you will have a tilted eyepieces, or in some cases the mechanical tube also might be tilted. So, you have a tilted binocular eyepieces, and you will have another port which will be for a trinocular port. So, what you have is, rather than having one, and you can see here very easily in this. Now, binocular eyepieces, the light will be from objective, the rays which are wave trains which are coming from the objective will be splitted by splitted and then enters the two eyepieces. The trinocular port, where you have three eyepieces, they could be used for different purpose. For example, the two persons would like to view the object and would like to view the image simultaneously, then, like for example, student and teacher, like teacher is either examining the student or teacher is explaining a particular sample or particular specimen to the student, then a trinocular port could be utilized. Another thing is that you can attach here a camera or a screen to permanently record the images, and that is why you can use a trinocular port upright here straight, and your other part is inclined for better viewing or for a convenient viewing in here. So, that is a very important part here, though we have made this figure in that way, but what you have is you have mostly inclined view in this. Now, we will discuss, like there is a coarse focus, there is fine focus, and this will be this is like you can adjust the microscope accordingly for focusing onto the specimen. Now, let us move on to the as we were talking about the two optical systems in the fixed tube lengths. Remember, fixed tube lengths is almost around 160 millimeter as accepted, widely accepted. Now, here in this figure, if you see, I have shown you the finite optical system. Now, finite optical system consists of objective lens, eyepiece lens, and then finally, you are viewing it directly through your eyes. What happens in finite system or finite optical system, which is also we called fixed tube length, the object is placed little farther from the focal length. Like you can see here, there is a object in the form of arrow has been placed. Now, this light, this illuminated object, the light falls on the objective lens and it is focused at the intermediate image plane. Now, this intermediate image plane will be placed in such a way that it is this is little less than the focal length of the eyepiece. Now, if you see here, the optical tube length which we were talking about, the mechanical tube length or optical tube length is from where the objective is threaded, and with it is like focal length of the objective on the other side and till the intermediate image plane, this distance is called optical tube length. Now, this acts as an object for the eyepiece, and eyepiece will make a virtual erect image on the same side of the lens, that is eyepiece. Now, this image is formed at a 25 centimeter or 250 millimeter from the eye lens, which is you can say a relaxed position for the eyes to view the objects. Now, this virtual image acts as the object for the eye lens, and finally, an inverted real image is formed on the retina of the eye. Now, which is of course, not perceived as the inverted image, rather it is perceived as the erect image as it is here by the brain. As we know that when we see the objects directly through our eyes, though inverted image is formed on the retina, but we see people as upright or erect. So, in finite optical system, it is very difficult to introduce auxiliary components between the objective and the eyepiece, because there will be problems of spherical or other aberrations, and the image formation will not be sharp. So, those to avoid those things, and many times you might have to increase the tube length also, mechanical tube length, which might not be possible. So, what advanced microscope now most of them contains, this is an infinity corrected optics. Now, in infinity corrected optics, what happens is that you are able to insert auxiliary components like a lot of polarizers, prisms are included in here, and without any image problem. Now, how does it work? It is a simple schematic here shows that what you have is, so you have other lenses like condenser lens system, you have object plane, and what you have is objective lens which is placed in here. There is an additional lens near to the objective lens, and it is called tube lens. Now, what here, the light coming from the specimen, that is diffracted light or direct light coming from the specimen, is directed at infinity by the objective. It is not focused actually, rather it is directed by the objective at infinity. And finally, so here objective is not really focusing the light, rather the tube lens is focusing. So, when it is directed to infinity, it falls onto the tube lens, and finally tube lens focuses the light onto the intermediate image plane. And so, in essentially when you have to determine the magnification of the objective here, you have to take two things. One is reference focal length, which is from tube lens till the intermediate image plane, and then you have to also consider the focal length of the objective, that is real focal length where you are focusing the object. So, magnification of the objective will be given by reference focal length divided by focal length of the objective. So, that is how you will be determining here. And finally, then as we have seen, let us see this in the ray diagram, as we it will be much simpler. Here you can see, like we have shown you in the finite corrected optics, here objective lens is there is a tube lens, and there is the space between the objective and the tube lens is called infinity focal space or infinity space. Now, this space is utilized for insertion of different auxiliary components, which you can insert in prism or polarizer or analyzer, whatever you would like to for your particular application, and that has made a lot of advancement and lot of variation in different types of microscope. And you can see that rays here which are coming, diffracted or direct rays which are coming from the object are focused at infinity, that is parallel rays almost are coming out in here, and these parallel rays are then, or infinity focused rays are then finally focused to intermediate image plane by tube lens. And like in other system, virtual image is formed here, which is erect and which is at around 250 millimeter or 25 centimeter as to say, and then finally eye lens will form a magnified image which is inverted on the retina, but upright on the perceived upright by us or by the viewer actually. So, you can see that there is a completely different, and this like here there is no image formation. Most of aberrations could be corrected in objective lens or they could be corrected in tube lens. Most of the time objective lens is corrected for aberrations, and it could be or it could be a tube lens. So, infinity corrected, now here one has to understand, it is not like there is an infinity space or something, it is infinity corrected only means that rays are focused at infinity, and finally they are brought to intermediate image plane by tube lens. So, modern microscopes most of them contains infinity corrected optics as we say. Now, supposing there is no direct viewing, like you are not observing directly the image, but you would like to record the image for permanent storage or viewing it later or analyzing it later, then little differences has to be put in here. You can see here the main difference is that earlier what we were doing is intermediate image plane was within the focal length of the eyepiece, that is your image formation, that is at intermediate image plane was smaller than the one focal length of the eyepiece, but now here it is little farther than eyepiece focal length. So, that now a virtual image will not be formed on the same side of the eyepiece lens. Rather, there will be a real inverted image, magnified image will be formed on the other side of the eyepiece lens, which could be collected on a screen or recorded on a video camera for further applications. So, projections can also be done, like in case of when you have a trinocular port and you are putting in camera in there, you would like to record the image rather than viewing there directly. So, this was little bit about different types, like we have seen infinity corrected or finite optic systems and different parts. Let us discuss these different microscope components in detail. And three main components which we are going to discuss in here. The most important is objective lens. Like I told you, a simple lens is made up of a single lens which is called objective lens, and it is called objective because it is closer to the object. Then second is ocular lens or eyepiece lens, which is closer to eyes where you are viewing the object. That condenser lens is the part of illumination system, and it focuses a fine beam of light onto the specimen in a very small cone, and we will see what kinds of illumination systems are available. If you could recall, in history we have told you there is a Kohler illumination. So, there is Kohler illumination and there is a critical illumination, we will discuss that. Then there could be optical filters, there could be diaphragms or as we say apertures, there could be photo adapters which are there, which are utilized for filming or in camera. Now, here we are going to discuss three of them, that is objective, ocular and condenser lens. Now, most of the objectives in the modern microscopes are standardized with respect to numerical aperture and magnification. Now, remember empty magnification is never allowed here, that is magnification is never empty, because you increase the numerical aperture as the magnification needs to be increased. Numerical aperture can be increased by decreasing focal length. If you could recall, we have discussed this, where numerical aperture is the light gathering capacity, and it could be increased by decreasing wavelength. You can increase it by not decreasing wavelength, that is a separate thing, but you can increase it by decreasing the focal length. You can bring the lens closer to the specimen, so that more light could be collected at higher angle. Now, also lens diameter decreases with increasing magnification. So, this also could be done. Then there are like we have discussed earlier, it could be oil immersion lenses or lens working in the air, or it could be water or glycerin. All of them has different refractive indices, and accordingly they will give you the resolution. So, high magnification in oil immersion is achieved by placing object almost within the lens, very close to the lens, since it has a very high numerical aperture. Because of this, in the oil immersion lenses there are residual chromatic aberrations, and those could be corrected by compensating eyepieces called apochromatic lenses. Also, objective lenses are corrected for spherical and other aberrations. There are flat field objectives which are, or plane objectives which are corrected for different aberrations, like flat field objectives are corrected for curvature of field. It is required for the microscope when you are doing photo micrography and you are covering the larger area. This figure shows a typical objective lens. As we can see here, that objective lens, the size of the objective lens is bigger than a single lens, because this consists of a series of glass lenses, and which are there to correct all spherical and other aberrations. Now, there are few things written in here which we should take a note of, and every objective will be inscribed by that. One is plane and apo, which is for corrected spherical and chromatic aberration. Then magnification, 60x here means that the image could be magnified 60 times, that is what magnification shows here. It could be 40x, it could be 5x, it could be 10x. The numerical aperture, if you see, that is the 0.95. Now, remember, as magnification increases, numerical aperture also has to increase to avoid the empty magnification. So, it could be right from very small to very high. Now, 0.95 is almost highest in the case of lenses which are being used in the air. This here tube length, if you see the infinity sign, shows that it is infinity corrected optics. This microscope contains, or this objective is infinity corrected. Another is, this is cover glass thickness is given. Many objectives will have a color code for different kinds of magnification, and there is a front lens which is very close to the specimen, and the working distance will be calculated from the front lens to the specimen where it is placed on a specimen stage. So, objective lenses, now if you compare certain properties of objective lens, like we are talking about, when you increase the numerical aperture, then you have to reduce the focal length. So, there are few things which we need to relate here. For example, when you increase the magnification as shown in this table from 10 to 95x, you can clearly see that numerical aperture also increases likewise, and but focal length decreases, that is, you are bringing the objective lens closer to the specimen. So, you are decreasing the focal length. Also the working distance decreases. The working distance, as I said, is the distance between the front lens to the specimen, and this will decrease as you increase the magnification and as you increase the numerical aperture. Diameter of field, that is the area which you can view, will also decrease, because farther you are from the specimen, more area could be seen. As you go closer to the specimen, less area you can see. We will just discuss what is diameter of field as such later on. So, this is about the objective lens, which are, like I said, very much standardized for magnification and resolution. Let us now move on to the next important part of the microscope, compound microscope, eyepieces or ocular lenses. Now, ocular lenses are also called projecting lens or projector lenses, because what happens is they are projecting the image which is formed already by the objective lens. Remember, objective is the main lens in the microscope, and the resolving power of microscope is solely determined by the objective, because objective collects the rays, diffracted rays, or most of the information directly from the specimen, and the eyepieces will collect the information from the image formed by the objective. So, there cannot be any additional information which eyepiece could collect. Only thing it can do is it could magnify the image to a certain level, so that more information can be viewed or observed. It is always better to have a higher magnification objective than a eyepiece. For example, 40x objective with a 5x eyepiece will give you much greater resolution than a 10x objective and 20x eyepiece. So, one has to remember that. There are different types of eyepieces, there are many different kinds of eyepieces, and we are not going to go in all of them, but some of the names here I have given which are used frequently, like Ramsden, Huygens, Kellner, and compensating eyepieces. Compensating we have discussed, like in oil immersion lenses for correcting the chromatic aberrations, residual chromatic aberrations, the compensating eyepieces are utilized. Now, Ramsden and Huygens eyepieces, which are widely used, and but there are many variations have come up. These two consist of two lenses at opposite end of a tube, that tube which is connected to the one end of the mechanical tube. So, what you have is two lenses, one is closer to the eye, which is called eye lens, and another is, or at the other end, which is called field lens. So, these two pairs of lenses are available with a diaphragm either in between the two lenses or at one end of the lens, as we will see in the figure. Now, if you consider Kellner lens, it is a modified version and of Ramsden with a doublet of eye lens, and it features a higher field of view for eyes, or you can say higher eye point and a larger field of view. Now, these are schematic of the two kinds of lenses which we were talking about. This is Huygenian lens. Now, you can see Huygenian lens, there is eye lens here, there is a field lens at other end of the tube, and in between there is a circular aperture opening which will determine the field of view, it is called diaphragm. Now, if you could see that the convex part, these are plano-convex lenses, and the convex part of the lens focuses onto the specimen side. These are also called, this is also called negative eyepiece. Now, in comparison, Ramsden eyepieces are also plano-convex lens, but if you could see that the convex part of the field lens faces the eye lens rather than towards the specimen. Also, the diaphragm is not situated between the two lenses, rather diaphragm is situated below the field lens, and this you can say you can mount adaptable for mounting reticles. Ramsden lenses are adapted for mounting reticles, and their focal length is kind of at the diaphragm here. So, these are called positive lenses, and they are used in many different applications. Hygienians lenses eyepieces are also used for in many teaching and research labs. There could be many other eyepieces which we are not going into. The third component is the condenser lens. Now, condenser lens are very important part of illumination system. Now, there are few conditions which needs to be fulfilled for object to be illuminated in an ordinary microscope. What are those conditions? One is that incident light must fill the numerical aperture to ensure the resolution of objective, that is the best resolution of the objective. So, first thing is that the numerical aperture should be completely filled to get complete resolution of objective. Second is the illumination should be uniform across the specimen, that is it should not be like the one part is illuminated more than another. So, it should be uniform illumination across the specimen has to be there to and one has to eliminate all stray light or noise. Now, condenser lens plays an important role to fulfill these criteria. So, what has to be done? One is that a small dimension source of light is focused down to a very small area by using multiple lens in a single lens unit called condenser. Now, there could be different types of condenser, 4 types of condenser lenses. One is Ebb lens, aplanatic, achromatic and aplanatic achromatic lens. Now, Ebb lens is not corrected for any aberration, they were very simple lens used earlier. Aplanatic lenses are those which are corrected for spherical aberrations, and achromatic lenses are corrected for chromatic aberrations. Aplanatic achromatic lenses are corrected for both aberrations. So, here these are 4 types, and as you have more features, they are more expensive also. Now, condenser can provide two kinds of illumination. One is called critical illumination, another is called Kohler illumination. Now, object is illuminated in let us discuss critical illumination. Object will be illuminated by a cone of light from a focused light source. Now, the position of the condenser with respect to the object is adjusted, so that to ensure that a spot of illuminating object is as small as possible. It is a cone, you can say small cone of light is illuminating the object. Also, it should be as bright as possible, it should be as intense as possible. And most of the time a diffused screen is put before the light source, like I told you. So, that the light source is not filmed here, not imaged here. Now, numerical aperture needs to be at least as great as the objective in order to fill the objective aperture. That is a very important part. If you cannot fill the objective aperture, you cannot get the required resolution of the or the full resolution of the objective lens. So, numerical aperture of condenser needs to be greater than the numerical aperture of objective. Now, this is a very simple schematic diagram shown here to show the critical illumination. It is a simple light source illuminating will be illuminating the object, and condenser will focus the light in a very small cone of area. Stray light has to be avoided through different apertures or diaphragms which are placed at different places, different sites, and a very small cone of light will illuminate the object at the object plane, and finally, the objective the diffracted and non diffracted light will enter the objective. So, critical illumination is a simple illumination, but now it has been replaced by Kohler illumination, which is much more intense and precisely controlled as compared to the critical illumination. Now, in this one there is an extra or accessory lens called field lens is placed between the light source and condenser, and when light rays leaves the condenser, it leaves as a parallel beam of light, many parallel beams of lights called pencils of light. So, you can say there are many small cone of light which are falling on the specimen at different angles actually. So, all light passes through the object at various angles, and the angle will increase with the distance of the source point from the optical axis. So, axis is here illuminated by sets of pencils that makes up the for the cone of light which was in critical illumination. Now, condenser can be adjusted, and it is made sure that stray light is avoided. If you see in this figure shows you a very simple schematic of how the Kohler illumination is achieved. You can see there is a field lens here, and this field lens, and there are field irises or substage iris at different places which will not allow the stray light to pass on. And finally, the object is illuminated rather than one cone of light, many pencils of light are illuminating the object uniformly, and this is much more precisely controlled, and you can get intense uniform beam to illuminate the object, and which is used in most of the microscopes. So, this was about critical and Kohler illumination by the condenser, and we have discussed the three kinds of lens systems which are important in here: objective, eyepiece and condenser. Now, there are few terms which needs to be discussed in here before we like move on to the particular microscopic techniques. One which, if you remember, with objective we were trying to discuss like field of view, other things. Before that, there are two terms, depth of field and depth of focus. Now, depth of field is a thickness of a specimen that can acceptably be sharp at a given focus level. So, that depth of field, if it is a particular thickness of a specimen can be at focus, then it is depth of field. So, once you go more than that, then focus is lost. Another term is depth of focus. Depth of focus refers to the range over which the image plane can be moved while an acceptable amount of sharpness is maintained. So, that is depth of focus. Field of view, which we were discussing earlier. Now, field of view is another important thing, that how much area of the specimen you are viewing. So, diameter of the field in an optical microscope is expressed by field of view number or simply field number. Now, this is the diameter of the view of field in millimeter measured in the intermediate image plane in most cases. Now, the eyepiece field diaphragm opening diameter determines the view field size. If you could remember, in Ramsden and Hygienian, we have shown that there is a diaphragm, that is circular aperture, and that will determine the field of view. Field size in specimen plane is then defined as the field number divided by magnification of objective. So, when we were say field view, that was an intermediate image plane, the field size it at the object or specimen level. The field size is given by field number and divided by the magnification, actually, that is objective magnification. So, you can determine the field size in the specimen plane by this simple formula. Another important part, like we have discussed, like resolution, magnification, but another important part of viewing an object or specimen is the visibility. Now, unless different components or different parts of the specimen are visible, that is, they could be differentiated, like different points in the object could be differentiated from each other and could be differentiated from the background, you will not be able to see. Lot of biological specimens are transparent, and many times though they could be observed, but they are very hard to be visible in the image, because the intensity differences between them, between the two objects in the specimen, or between the object and the background is too low. So, they could not be seen. Now, here comes the term contrast. So, there has to be contrast for visibility. What is contrast? Contrast can be defined as the difference in light intensity between the object or image and the background. Now, here the difference in color and brightness of an object from other objects in vicinity and background creates contrast and allows the details of an object to be visible. For human eye, minimum contrast value is of 2%, that is 0.02, which is required to distinguish the differences between an object and its background, and it could be given by a simple formula, that is percent contrast could be calculated by IS, that is the background. IS is the specimen intensity minus IB, which is background intensity, into 100 divided by IB, which is the background intensity. Like I said, most biological specimens are transparent and are not visible because of they lack contrast, and there has to be created contrast by different techniques. One is staining the objects with particular dyes which them a particular and those dyes can absorb light and can give intensity differences. But there are other methods also which are also utilized for creating contrast. Like we are going to discuss in the coming classes in the or in the coming lectures, how the contrast could be created without staining the objects. Now, when you stain the object or specimen, then what you do is you to certain extent you cannot observe live cells or the cells as it is. There will be some differences in observing live cells and the stained cells. Other than staining, there could be different optical phenomena could be used to create contrast. For example, we will be discussing dark field microscopy, phase contrast microscopy, fluorescence microscopy, polarization microscopy, for and interference microscopy to see how contrast is developed in these different techniques. And then also we will be discussing how specimen is stained, how these samples are prepared for the observation under a microscope. So, in the next lecture we will be discussing specific techniques, and we will start with dark field and phase contrast microscopy. Thank you very much.